everybody. We're going to talk about RNA transcription today. So in the past, we talked about DNA replication, and we talked about how DNA replication is necessary when you need to make a new cell, when you're either a somatic cell, so that would be something that's in your body like skin, liver, muscle, hair, teeth, those sort of cells. When they need to replicate themselves, when they need to make an identical copy, they have to replicate their DNA too. They have to take with them a copy of their entire library on how to make proteins. This is also true for cells that are involved in reproduction. So the germ cells, these are cells that make sperm and egg. Even though these cells go through meiosis, a completely different type of DNA, different type of cell replication, they still need to duplicate their DNA first before they switch alleles over and do all that cool stuff, and turn into haploid sperm or egg. But regardless, that's DNA replication. And now we get a chance to kind of look at the other aspect of DNA. And so that other aspect of DNA is that it's a repository or a library of information on how to make a protein. And so the best way to think about this is that DNA is like a giant library full of recipe books. And these recipe books tell you how to make all different kinds of proteins. And we say that, sorry, this cat's really distracting. Hold on a second. And so that library, the DNA library, stores all this information on how to make proteins. Now, this is a parallel look at how DNA functions, right? The function of DNA. DNA is regulated, especially in eukaryotes, in a very sophisticated way. We kind of simplify it by saying, oh, there's a recipe. The recipe codes for a protein, but it's a little more complicated than that. So remember that the sequence of nucleotides in a gene, so that's the order and sequence of nucleotides, A, T, C, G, is paralleled to the order and sequence of amino acids. Now, it's not a one-to-one exchange. It's not just like, oh, A is equal to tryptophan, G is equal to lysine or something like that. It's actually the translative aspect from the order and sequence of nucleotides to the order and sequence of amino acids is done in a 3-to-1 way. So 3 nucleotides are going to code for 1 amino acid, but they run in parallel. So it's every 3 nucleotides, 1 amino acid. The next 3 nucleotides, the next amino acid. The next 3 nucleotides, the next amino acid. But anyways, let's just talk about proteins. So DNA serves as a repository for proteins. Let's remember that proteins have many functions in the body. Okay, so one of those functions of proteins in the body is that we talked about last unit was enzymes, right? So they're able to break things down, build things up. They're unchanged by the reactions that they facilitate, just like a catalyst, right? They're a catalytic protein. We also have structural elements. So these are proteins that make up like the cytoskeleton. They help to support the cell. They're also going to provide like highways, if you will, for transporting different molecules throughout the cell. We'll look at transmembrane complexes, or a different type of protein. We did talk about these in depth in Unit 1, but they're proteins that are embedded into membranes in the cell. So these facilitate transport, they transduce signals, they facilitate enzyme reactions, they help join other cells to other cells, like your skin cells, right? They offer channels for ions to go through, and they also can act like pumps. DNA are proteins that come from DNA, which is where they all come from, can also serve as hormone signals. So if a cell needs to talk to another cell and it doesn't have a phone, it can send a protein signal through the bloodstream, and the other cell will hopefully have a receptor for that message, and then they can communicate with each other. And then finally, pigments. So pigments would be like skin color and things like that. Eye color, skin color, hair color, anything that has color that's in the body is made from a pigment. Even just like the pinkish color of your liver and your stomachs and stuff. If you decide to disembowel somebody, you can see that there's pigments all over the body, and those are all from proteins. So let's take a check. And so remembering the amino acid in the protein is parallel to the nucleotide sequence in the gene. We call the sequence of nucleotides in DNA that code for a functional product a gene. And so here we can see we can actually take regions of DNA from one organism and insert them into the DNA of an other organism, and that other organism will express those genes and express those proteins as if they were that other organism. So we have a tobacco plant that's glowing in the dark like a firefly because we took some firefly DNA and inserted it into the tobacco genome. The pig is expressing a jellyfish gene in this picture. So it's just showing you that DNA is interchangeable. We can take certain parts of DNA from one organism and put it in another, and it's functional in some cases. So let's talk about, we know what a gene is. We can skip that slide. So in prokaryotes, um, they're going to have less regulation over their DNA and less regulation over transcription and translation simply because they don't have a nucleus. There's no chance for post-transcriptional modification just because the RNA transcript is going to get translated right away. There's no membrane in between the processes that are happening in this image. It's just DNA gets transcribed, translation happens immediately afterwards. But in eukaryotes, this is where it gets interesting. In eukaryotes, we have more compartmentalization of our DNA and its regulation. So we have transcription taking place in the nucleus, but while it's in the nucleus, it can go through post-transcriptional modification. So think transcription is just taking DNA, making a photocopy of its recipe. Post-transcription happens after transcription. So post-transcriptional modification means we can modify the RNA transcript, modify the recipe to suit our needs. So what often happens when you have this ability to compartmentalize your reactions is that the DNA will get turned into RNA, and the DNA, let's use the example of the recipe. Sorry, I get distracted by this cat that's like all over me. So let's use the example of the recipe. So DNA is a recipe. Let's say that this gene that we're looking at here in this image is a gene for oh my gosh, cat. Here, just go. Let's say that it's a recipe for like a cake, something that's super sugary. It's got frosting and all kinds of stuff. So we take that recipe. Oh boy. So we take that recipe and we are going to make a copy of it. So we make that copy into RNA. And then let's say that maybe we don't want a sugary cake, but we can still use all those ingredients and use most of the process. What we're going to do is we're just going to cut out, so this kind of lighter peach color, we're going to cut that part out of the recipe, and then we're going to glue all the other ingredients and other steps back together. And then now what we have, instead of a big cake, is we have the recipe for a piece of bread or a loaf of bread. And so it's what's really cool is that post-transcriptional modification allows so much more flexibility in the products that you can make. Yeah, so it's pretty cool. So let's take a look at this DNA molecule. All right, so here what we see is we remember that there's two strands in a DNA molecule, right? They run anti-parallel to one of them. Only one of them is actually read by the RNA polymerase. So the RNA polymerase is going to read the template strand, but it's going to make a copy of the template strand that looks like the other side of the DNA molecule. Other than that, it's going to look just like the other side of the DNA molecule, but it's going to be an RNA. So it's going to have uracil instead of thymine, and everything else will be the same. That is called the template strand. The RNA copy it produces looks identical to the sense strand. So we can see that on the next page. So the RNA copy produced looks identical to the sense strand, other than it's got the uracil instead of thymine. So let's look at, we'll look at RNA polymerase. So this is RNA polymerase reading the template strand, which is running 3' to 5'. So it's reading 3' to 5'. And then notice that the RNA strand it's synthesizing is in an anti-parallel direction. So it can only add new nucleotides onto the 3' end. So it can only add this A onto this other A, because that's the 3' end. So it can add a new nucleotide onto the 3' end. So the strand it's synthesizing is being synthesized in a 5' to 3' direction, just like the sense strand across from it. So we can see that on the sense strand, it reads CAT, but on the RNA transcript, it's reading CAU. Yeah. Moving on. So what we have here is we have a diagram that you're going to see on your test. And so I want to talk about each region and then what it's representing. Okay. Region A here, that's the promoter region. So the promoter region is a specific sequence of DNA just before the beginning of a gene. So the promoter region is not transcribed, but the nucleotides themselves serve a function. And so inside of the promoter region, you're going to have a TATA box And you're also going to have other nucleotide sequences that attract proteins called transcription factors that are going to bind to the DNA and kind of open it up and get it ready for the RNA polymerase to come in and start transcription. So that's area A. That's this area. So this would be your promoter region. The 3' end of the gene would be over here on the left on the top. The 5' end would be over here. And this top line would be considered to be the template strand. The bottom line down here is going to be the sense strand, and it's going to be anti-parallel to the template strand. And so it's going to be anti-parallel to the template strand, and it's going to go from 5' over on the left to 3' over on the right. RNA polymerase, the transcription factors orient, the C right here, the transcription factors bind to a sequence just before the start of the gene, and those transcription factors can either turn on the gene for transcription, they can either open it up and allow the RNA polymerase to come in and start transcribing DNA into RNA, for sure, or they can switch the gene off. They can actually keep the strands together, keep RNA polymerase from actually doing its job. And you might say, well, why would you ever want to do that? Well, consider that maybe there's a 4-dimensional aspect to how your body operates. Maybe there's certain genes that it needs switched on at certain times of the day. Maybe it has certain genes that need switched off at certain times of the day. And so this is the way that the body can actually turn on or turn off a gene. So it creates a 4-dimensional aspect to the gene. It's not just like, oh, the gene gets turned to RNA, RNA becomes a protein, protein's functional, end of story. It's more like, no. DNA creates RNA, creates protein. Protein is a transcription factor that then goes and sits on another gene, turns that gene on or off. That gene could also be a transcription factor that goes and turns on or off another gene. So there's this 4-dimensional web of interactions between the proteins that are created by DNA and the DNA itself. All right, so we talked about the promoter region. The TATA box is an adenine and thymine-rich sequence of DNA that comes just before the gene. And you might say, well, why would you want a sequence of DNA that's just A's and T's? Well, consider this, right? You remember that adenine and thymine make 2 hydrogen bonds between the 2 strands, right? If adenine's on one side and thymine's on the other, they bond together with 2 hydrogen bonds. So that's like having 2 magnets zipping something together. Cytosine and thymine have 3 hydrogen bonds. And so that's like 3 magnets holding each other together. So if you have a gene that you want to transcribe, you have to separate those 2 strands of DNA from each other. That separation is a delta G positive event, meaning it's endergonic, meaning it requires the input of energy from an outside source. Well, if let's zoom out and look at the macroscopic picture. If you're an organism who is struggling for survival, most likely you're struggling to find food, and that food is your energy supply. Why would you waste energy opening up 2 strands of DNA if you could do it easier with A's and T's than C's and G's? And so the TATA box creates an easier, less energetic way of opening up the DNA. It's a conservation of energy. We talked about transcription factors that can turn on and off a gene. The RNA transcript itself, oh no, I exited the slideshow. The RNA transcript itself is the photocopy of DNA, but in RNA form. So that's what the RNA polymerase itself is making. An RNA polymerase is the enzyme, right? So it is a protein itself that is responsible for copying the DNA into RNA. The antisense strand is the template strand that is read by the RNA polymerase. And then the sense strand is the opposite side to the sense strand, or to the antisense strand. The sense strand looks exactly like the transcript being made, but again, the U's are there instead of the T's. And then at the end, so you might wonder, okay, so we have all this machinery to get the gene transcribed. Then you might wonder, well, what keeps the RNA polymerase from just going forever? Well, at the end of the gene, there's something called a polyadenylation signal, or polyadenylation signal sequence. And so this is a specific sequence of nucleotides after the gene that tells the RNA, like when it reads it, like it's a pattern, it's a code. And as soon as the RNA like comes into contact with that code, it's like, oh, there's my signal. I can just pop off. I'm done. My job's over. So that's a pretty cool way to regulate that. All right. So now that we have an RNA transcript, now that the RNA polymerase is let's start over. So now that the promoter gene has been The promoter region has been interacted with by transcription factors. Maybe they've switched the gene on, opened it up at the TATA box. RNA polymerase comes in, gets oriented in the right direction on the template strand and starts copying. Reaches the polyadenylation signal, falls off. Now we have an RNA transcript. We have an RNA photocopy of the DNA, a photocopy of the recipe. Now we can go through post-transcriptional modification. And remember, this is taking place inside the nucleus, and so it's this compartmentalized way or reaction, a way to isolate what's happening to the RNA transcript so that we can make these changes to it so that it can become, we can become more sophisticated in the products that can be made from this transcript. So two things that will always get added to an RNA transcript is a cap at one end, and it's made by guanine triphosphate. So if you remember, to build a polymer is going to require the input of energy. It's going to endergonic reaction. So if we place a little battery at the beginning of the RNA transcript, that's going to power the ribosome to get started with translation. At the opposite end of the transcript, at the 3' end, we're going to just enzymatically place a whole bunch of adenines onto the end. And so they're added onto the end so that when this RNA transcript is finished and ready to be translated, when we send it out of the nucleus into the cytoplasm, the cell's immune system, and it does have its own built-in immune system, if it starts to attack that RNA transcript, right, because it sees it and it's like, wait a second, this could be viral, it could be a virus, I want to get rid of it. So if it starts to attack in it and it's eating it from the 3' end working towards the 5' end, it's just eating a bunch of A's. It's just like yomp, yomp, chomp, chomp. It's like Pac-Man working its way up. And hopefully the ribosome gets to the RNA transcript in time to translate the protein before those enzymes eat the poly A tail. Something to consider too, it's kind of fun, right? Is that if that poly A tail is long enough, that you can change how many times that gene is actually translated. So a ribosome is going to come on and translate it. It's going to turn into a protein, and when it does, it might not be done. It might start translating, and then another ribosome hops on it right after it, and then another ribosome right after it. So you might get multiple copies of a protein from one RNA transcript. So if you can extend the poly A tail and make it really long, you can get a bunch of proteins from that one copy. Or if you make it shorter and you don't need as many proteins, you can reduce the amount of proteins that are made from that copy. So again, these post-transcriptional modifications can create a lot of diversity in the cell. And then finally, this is the best part. This is my favorite part, spliceosomes. They're part protein, part RNA complexes. And what they are going to find is they're going to look on that RNA transcript, and because they have RNAs, like small RNAs inside of them, those small RNAs inside of the spliceosome will target regions of the transcript and cut them out. And those regions that it targets is called an intron. It's in there and it needs to get out. The exons are outside of the introns and they need to come and get glued together. So the spliceosomes cut out introns, glue together exons. In some ways, sensibly, it seems almost opposite, right? Like the terminology just seems opposite. Like you would want to keep in the introns and X out the exons. But for some reason, somebody named it the other way. So we cut out the introns, splice the exons together. That's what gives us our recipe. So I'm going to show you a model of that. Here we have our mRNA transcript. It's not mature yet, though, so it's more just an RNA transcript. It's got a polyadenylation signal, which causes the RNA polymerase to fall off. That polyadenylation signal is going to signal another enzyme to come in and add a bunch of A's, a bunch of adenine to the tail, the 3' end. That's the poly A tail. The GTP cap gets added to the 5' end, so it's got these 3 phosphates that are going to power the ribosome to get it started. And then you have a little bit of a region that UTR is untranslated region. So that's a part that's not going to actually get turned into an amino acid. Then you have your start codon. So that's where the sequence of amino acids is going to parallel the sequence of nucleotides in this transcript. inside of the actual region that codes for the protein segments, you have these peach-colored segments that we called introns on the last slide. Now, those introns are going to get spliced out by the spliceosome. So this image of a spliceosome is just obviously like a diagram. It's probably not what it looks like in real life. But notice that inside of the spliceosome, it's got little RNAs that are going to target parts of the intron. They're going to say, oh, here's the beginning of the intron. Here's the end of the intron. Let's cut that. And then the protein, the enzyme itself, the spliceosome, cuts the intron and glues the two exons, exon 1 and exon 2 together. So intron gets removed. Get back to my pointer. This intron gets removed by the spliceosome, and then the two exons, exon 1 and exon 2, get glued together. So what it started as is it started as this big, long product. After transcription, we post-transcriptionally modify it, add a cap, add a tail, splice out the introns, and now we're left with a mature RNA or messenger RNA, mRNA. And mRNA is what's going to go to the ribosome. Okay, and my cat just totally jacked up my slideshow. And that mRNA is what's going to go to the ribosome, and the ribosome will do its translation. All right, hope you enjoyed that lecture. I'll see you guys on the next one.